Hi there, everyone. We are so happy to have you, whether you are joining us today or will be watching later on the Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions in the chat box below. We will have time to answer questions at the end of the presentation. My name is Monica Joanna Almacabe, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trial. Today, we welcome Dr. Nazanin Mohammadi, who recently defended her PhD thesis, focusing on the clinical and functional characterization of GABA-A receptor-related disorders. Under the guidance of Professor Rikke Møller and Associate Professor Ahring, Dr. Mohammadi's research has made clear the complexities of gain-of-function and loss-of-function GABRB2 disease. Her groundbreaking findings reveal that GABRB2 variants can lead to both types of receptors with associated phenotypes strongly linked to functional outcomes, particularly in epilepsy. Dr. Mohammadi's research sheds light on the intricate nature of genetic variants in GABA-A receptor, enhancing genetic counseling and paving the way for personalized treatment strategies. I'm handing it off to you, Dr. Mohammadi. Please explain how the unraveling is of gain of function versus loss of function in GABRB2.
Hi, Monica. Thank you for the introduction, and hi, everyone. Thank you for joining today. I am happy to be able to present the project that I have been working with during the 3.5 years of my PhD. And the title is the Clinical and Functional Characterization of GABA-A receptor-related disorders, translating genetic diagnostics into personalized treatment. For today, I will go through a brief introduction, our aims, methods, results, and conclusions. And GABA-A receptors, they are members of the cis-loop receptor superfamily of pentameric ligand-gated ion channels. They respond to major inhibitory neurotransmitter GABA in the CNS, mediating phasic and tonic inhibition. Each fully functional receptor consists of 5 subunits. In most of the cases, it's usually 2 alpha subunits, 2 beta subunits, and a gamma subunit. As we see in the pie chart, there is different and a lot of different subtypes of this receptor, and the most common one is the one shown in the blue part of the pie chart, which is alpha 1, beta 2, and gamma 2. This subtype is expressed almost through all the brain, and here we see the receptor seen from above and in the cell membrane. It has 2 alpha subunits shown in blue, 2 beta subunits shown in pink, and a gamma subunit shown in purple. In this project, my aim was working with the beta 2 subunit of this receptor, but then later we got interested in other subunits as well that I explain later. As we all know, brain function relies on balanced inhibitory and excitatory activities, and any changes in GABAergic signaling can lead to major disorders, such as epilepsy, developmental delay, intellectual disability, movement disorders, and others as shown in this figure. Pathogenic variants in genes encoding GABA-A receptor subunits have been associated with spectrum of epilepsies ranging from self-limiting epilepsies to severe developmental and epileptic encephalopathies. And how can these variants affect the function of the GABA-A receptor? As we see in this figure, there are different levels that a variant can affect the function of the receptor, such as the sensitivity of it to GABA or subunit or receptor expression and desensitization that we will talk about more in the next slides. And epilepsy was the major focus of this project, and it's the most common chronic neurological disorders affecting more than 50 million people worldwide. It's a spectrum of a disease. It has different causes, severities, epilepsy syndrome, seizure types, and comorbidities. As many of you are familiar with the International League Against Epilepsy, which provides guidelines for classification of epilepsy, considering the seizure types, epilepsy types, comorbidities, and etiology of the epilepsies. In the past, the majority of epilepsies, known as idiopathic epilepsies, clinicians didn't know the cause of the epilepsy, while some of the epilepsies were caused by stroke, trauma, infection, and other known factors. But later on, it was discovered that 70 to 80% of these epilepsies were genetic, either polygenic epilepsies or monogenic epilepsies. In this particular project, I was focusing on monogenic epilepsies. Using next generation sequencing for the diagnosis of epilepsy in the past 10 years have led to increasing number of identified genes associated with epilepsy, and it's increasing by each year due to the lower price of the procedure and higher speed. And how do we benefit from genetic testings is that it gives the patients a clear diagnosis and save them from spare additional diagnostic procedures, gives better knowledge on the prognosis, treatment options, and it can help creating patient communities where families and relatives can communicate about the disease and help and support each other. Increasing number of the patients with genetic diagnosis, the demand for precision medicine is also increasing, which targets the defect molecular mechanism underlying the disease. The precision medicine considers the clinical symptoms, lifestyle, and genetic and other biomarkers in the patients. As shown in this slide, there are already precision medicine for some of the gene variants, such as SCN1A or SCN2A and other genes. When it comes to GABA-A receptor subunit variants, we only have knowledge about GABRB2, GABRB3 gain of function variants, and the recent studies suggested that patients with gain of function GABRB2, B3 variants should avoid GABAergic drugs. And functional testings are one of the initial steps toward identifying precision medicine. So in case of GABA-A receptor subunit variants, electrophysiological recordings have been used to functionally testing these variants. And initial studies have identified loss of function, referred to as LOF, as the underlying mechanism for the variants associated with epilepsy and other neurodevelopmental disorders, such as variants in alpha 1, alpha 5, B2, and B3 subunit of the receptor. When we were looking at the patients with GABAA-B2 variants, they had distinct clinical phenotypes, as I will present here. Here we have two patients with GABAA-B2 variants. One is mild and one is severe. Just to illustrate that patients are affected by these variants in different magnitudes. Some are very severely affected and patients die with, but some others can continue living but having complications. That's why we thought that loss of function as the only underlying mechanism cannot explain these clinical differences between the patients. So for this purpose, we wanted to actually characterize presumed pathogenic GABAA-B2 variants and explore genotype-phenotype correlations. We would also like to investigate whether variants alter the sensitization property of the receptor and how this parameter contributes to the underlying phenotype of the patients. And then we performed a second study where we looked at investigating if different amino acid substitutions of highly conserved transmembrane domain 1 proline amino acid residue, which is very important for the function and gating of the receptors, can lead to the same functional outcome across different subunits of the receptors. And basically, we would like to look at the paralog genes and see if different substitutions at this specific amino acid would lead to the same functional outcome. Moving on to the methods and how we performed these studies. For both studies, we gathered published and unpublished patients with GABA-A receptor variants, and we collected their clinical and genetic information and registered in REDCap database. We looked at epilepsy syndromes, seizure types, intellectual disability, movement disorders, response to medication, and other impairments such as vision, language, and sleep. And for the functional testing part of the study, for both studies, we performed the electrophysiological measurements using 2 microelectrode voltage clamping in Xenopus lavis oocytes. And the parameters that we had focused on was the GABA sensitivity, maximum amplitude current, and the sensitization, as we mentioned in the beginning. And here's a picture of the laboratory, electrophysiological laboratory, and the oocyte itself, which is being poked with 2 microelectrodes in the lab. And as just mentioned, we used oocytes, which are frog eggs, as our cell models for these studies, and they were injected with wild type and mutant receptors, either heterozygous or homozygous for the variants. For this purpose, we use concatenated cDNA construct, as illustrated in this figure here on the right side. We have 2 beta-2 subunit position, 2 alpha-1 subunit position, and a gamma-2 subunit position. In the study 1 that we were investigating GABAA-B2 variants, we subcloned the GABAA-B2 variant into one of the beta-2 subunit positions. And this was with respect to Mendelian distribution that recommends that there is a chance, 50% chance, that we will have single mutant receptors. In the second study, in addition to electrophysiological measurements, we also did cell surface expression analysis using TSA-201 cells, which were transfected with wild-type tagged subunits and proline mutation harboring tagged subunits and analyzed by ELISA. So in the summary, we were able to gather 26 missense variants in GABAA-B2 gene, carried by 42 published and unpublished cases. We performed electrophysiological recordings, genotype-phenotype correlation analysis, and for study 2, we had 7 transmembrane 1 proline missense variants in alpha-1, beta-2, beta-3, and gamma-2 subunits, harbored by 11 patients. Again, electrophysiological recordings, and in addition, also cell surface expressions. Looking at the results from study 1, where the main focus was GABAA-B2 variants, we looked at GABA sensitivity, and in this graph, we see changes in GABA sensitivity between wild type and GABAA-B2 variant receptors. On the y-axis, we see delta log EC50. On the x-axis, we see the variants and the wild type. The red bars, they illustrate the variants that decrease the GABA sensitivity of the receptors compared to the wild type, and they are referred to as loss of function variants. Purple bars, they are representing the variants that increased sensitivity of the receptors to the GABA compared to the wild type and identified as gain of function variants. There is one variant shown with the gray bar that didn't change the GABA sensitivity compared to the wild type and referred to as a neutral variant. When we look at the purple bars, gain of function variants, we see some of them, the red borders. They are variants that are loss of function in terms of current that we will talk about in the next slide. Another important message from this graph is to consider the fact that gain of function variants, they are increasing the GABA sensitivity compared to the wild type in different magnitude. So some of them are changing it with the low shift and the other one with high shift. So within the gain of function cohort, we will have low shift gain of function subgroup and high shift gain of function subgroup. And this is according to the phenotype of the patients that we will also talk about later. Another parameter was maximum amplitudes shown in this graph on the y-axis, and on the x-axis we have the variants and the wild type. There are 7 variants that decrease the maximum current of the mutant receptors compared to the wild type. 4 of them, shown in red, they are already characterized as loss of function in terms of GABA sensitivity. And the remaining 3, shown in purple, they are identified as gain of function in terms of GABA sensitivity. So these receptors have a mixture trait of gain of function and loss of function at the same time, meaning that they are highly sensitive for GABA, but the amount of receptors expressed in the cell surface are less than the wild type. And this made us believe in the beginning that this could protect the patients from having severe phenotypes, since they have less gain-of-function receptors on the cell surface. But since a cohort of the patients harboring these variants were too small, so we couldn't conclude for sure what is the effect of loss of current. So we just moved on with the gain-of-function cohort, loss of function cohort, and divided gain-of-function cohort into high shift and low shift gain-of-functions. So we moved on with the genotype-phenotype analysis, having a loss of function cohort with 13 patients, gain-of-function cohort with 27 patients. And as mentioned, we divided the gain-of-function cohort into low shift gain-of-function and high shift gain-of-function. We looked at different clinical parameters such as their gender, epilepsy types, syndromes, seizure triggers, and many other parameters. The overall message of this study was that patients in loss of function cohort had milder phenotypes compared to the gain of functions. And when we looked at the gain of function cohort, and we saw that in the high shift gain of function cohort, patients were even more severely affected compared to the low shift gain of function cohort. There are some clinical features that I have highlighted with the yellow that we will look at in the next slides. One of the first features were median age of seizure onset, shown in months. Loss of function cohort, patients had the median age of seizure onset at 7.5 months, while it decreased to 4.5 in gain of function cohort. Looking at the different subgroups of the gain of function cohort, patients in low shift gain of function cohort had median age of seizure onset at 8, while the age of seizure onset was 0 months, 7 days in high shift gain of function cohort. Then we looked at the fever sensitivity, shown mainly in, which is observed mainly in loss of function cohort with 90%, and decreased to 14% in gain of function cohort, and distributed differently in low shift and high shift subgroups. Then we looked at movement disorders, including dystonia, dyskinesia, hyperkinesia, and chorea. Looking at the distribution, we see that it's a feature that belongs only to the gain of function cohort, and the percentage of the patients having movement disorders increased also within the gain of function cohort belonging to high shift gain of function cohort, subcohort. Then we looked at neuropsychiatric features such as ADHD, autistic features, anxiety, and other features. And here we see that highest percentage with 82% belongs to loss of function cohort, and it falls to 43% within the gain of function cohort. Patients in high shift gain of function subcohort, they had the lower percentage of neuropsychiatric features compared to the low shift gain of function cohort. This can be explained by the fact that patients in the gain of function cohort are already severely mentally disabled, where performing psychiatric evaluations can be very hard. So we see this feature mainly in the loss of function cohort. Then we looked at the microcephaly, which was only observed also in gain of function cohort with the highest percentage belonging to the high shift GOF subcohort. And early mortality within 4 years of age was also a clear marker for the gain of function cohort with highest percentage in high shift gain of function subcohort. And looking at these different parameters, we would like to see if we can use these clinical parameters to predict if a novel GABAA-B2 variant is a loss of function or gain of function using our clinical markers. And the answer is yes. If we have an infant or newborn with seizure age of onset before 3 months, having early infant or early onset and infant syndromes with key features, severe feeding difficulties, having additional features such as movement disorders and microcephaly, That's a clear indication of high shift gain of function variant. And if the age of seizure onset moves to after 3 months, and the patients have syndromes such as neurodevelopmental delay and DEE, with additional features such as movement disorders and microcephaly, then it's an indication of a gain of function variant, while moving with the seizure onset after 3 months, having syndromes such as GOF plus spectrum, key features such as fever sensitivity. These are clear markers of a loss of function variant. Then later on in the school age, we can look at intellectual disability, where severe intellectual disability is a marker of a gain of function variant. Mild to moderate is indication of a loss of function variant. Looking at mobility and language development, being non-ambulant and non-verbal are clear indications of gain of function variance. Being able to walk and being able to talk are clear indications of loss of function variance. So at the end, the key message was that GABAA-B2 variants can be loss of function variants and gain of function variants. Loss of function variants are associated with fever sensitivity, mild to moderate developmental delay and intellectual disability, psychiatric and behavioral features, while gain of function variants are associated with younger seizure age of onset, moderate to severe DD and ID, microcephaly, movement disorders, and early mortality. So as I mentioned, we had divided the gain of function cohort into high shift gain of function variants and low shift gain of function variants. And patients within the low shift gain of function variants had milder phenotypes compared to the high shift gain of function variants. But we had one boy from Australia, a 4-year-old boy with seizure age of onset at 5 months, diagnosed with neurodevelopmental delay and epilepsy, daily seizures, severe developmental delay. He was nonverbal and non-ambulant with movement disorders, chorea, and dystonia. And unfortunately, at age of 4, he deceased from sudden unexpected death in epilepsy. So we would like to see if he was, we were expecting him to be mild in the phenotype, but he's severely affected. So we would like to see if other parameters also contribute for his severe phenotype. So for his case, who had L255V, which was a low shift gain of function variant and associated with this severe phenotype, we would like to go into the details. And for his case, we created receptors having the mutation in one of the beta-2 subunit positions, each individually, having 2 single mutant receptors and a receptor where the mutation is subcloned in both of the beta-2 subunit positions. We started by looking at the GABA sensitivity as we did for the other variants for all 3 receptors, and we see that in all cases, his variant will cause a gain of function receptor. And looking at the current amplitudes, we see that all the receptors had the same maximum current amplitudes, meaning that his receptors in all case scenarios will have the same chance as the wild type to express the receptor to the cell surface and leading the current passing through the cell. This contributed for sure for his severe phenotype. Then we moved on to desensitization property changed by his variant. We looked at decay constant and steady state currents. 2 of the receptors, they increased the desensitization rate, and at the steady state current, we see that 2 of the receptors increase the steady state currents, meaning that 2 of the receptors were able to increase the desensitization of the receptor, meaning that the receptor was quickly desensitized and ready for another activation. which was clearly also another contribution to his severe phenotype. So we moved on to the study 2, and here we would like to see one specific amino acid, which was the transmembrane domain 1 protein, conserved in all the subunits of the receptor. And for this purpose, we found the 11 patients having this mutation in alpha-1, beta-2, beta-3, and G2 subunit. And I'm not going to go into the detail of the patients as shown in the slide, but the main message was all of them were severely affected with intractable seizures, early onset syndromes, with severe ID and DD, with a lot of comorbidities. We moved on with functional testings. As in study 1, we looked at GABA sensitivity, and for this study, we also created the 3 types of receptors having the mutation in one subunit and the other subunit, and in both subunit positions. When we looked at the data for GABA sensitivity, what we saw was that all the variants in all subunits led to a gain of function trait, and we moved on with maximum GABA activated current amplitude, as we saw in study 1. Here we saw that when the mutation was cloned in both positions of the alpha-1 subunits, beta-2 subunits, and beta-3 subunits, all the different amino acid substitutions led to no change in maximum current, beside the ones that were subcloned in both the positions, meaning the homozygous receptors for the variants. And when we looked at the substitutions, 2 substitutions in G2 gene, GABAA-G2, then we saw that the maximum current wasn't decreased or increased by these variants. And as we were expecting that loss in the current would protect patients from severe phenotype, that wasn't the case. Actually, all patients had the same and very similar phenotypes. So loss in the current wasn't a strong contributor for the phenotype of the patients. Then we moved on with the desensitization properties altered by these variants. The main message was that M1 protein substitutions with other amino acids in all subunits led either to increase or decrease the desensitization, while when the mutation was cloned in the mutations in G2 subunit didn't alter desensitization. So we were expecting that changed desensitization will either protect the patients from severe phenotype or worsen their phenotypes, but this wasn't the case either, and this wasn't identified as a main contributor for the phenotype of the patients, since patients having the mutations in G2 subunit had the same phenotype as patients having the mutation in alpha-1, beta-2, or beta-3 subunits. Then we moved on with the cell surface expression analysis. Here we have introduced the mutation, proline to leucine mutation in alpha-1 subunit and in beta-2 subunit. And what we see overall by this analysis is that the proline mutation decreases both alpha-1 and beta-2 subunits express cell surface expression and whole cell expression. And at the same time, both in alpha-1 study and beta-2 study, it also decreased the overall receptors expression. And since the cell surface expression and whole cell expression is decreased in the same magnitude, that means that these variants were able to decrease both cell surface expression and whole cell expression levels, meaning that it's not a trafficking issue of the receptors to the cell surface, but maybe more of a receptor assembly or transcription issue here. And we haven't performed these studies for other subunits, but again, having less cell surface expression, we were expecting that this will protect patients from a severe phenotype, but that wasn't either the case because patients with this mutation in beta-3 subunit or in G2 subunit, they had the same phenotypes as patients in alpha-1 and beta-2 subunit. So we looked at all the parameters that we have measured and the phenotype of the patients in alpha-1, beta-2, and beta-3 and gamma-2 subunit, where they all had severe phenotypes. When we looked at the GABA sensitivity, it identified all the variants as gain of function. When we looked at the maximum current, we saw loss of function only in the homozygous receptors. And this wasn't the case for the GABAA-G2 variants, since there's only one G2 subunit in the receptor. Desensitization was changed, either increased or decreased in alpha-1, beta-2, and beta-3 subunits, but not in G2. And cell surface expression was decreased in alpha-1 and beta-2, and not tested in beta-3 and G2. Looking at all these parameters measured, we see that GABA sensitivity is the strongest parameter to characterize the severe phenotype of the patients. And as mentioned, they are all gain of functions with similar and severe phenotypes. So GABA sensitivity is the most strong parameter contributing to the severe phenotype of the patients. So what we concluded from two studies was that GABAA-B2 variants can cause both gain of function and loss of function effects associated with distinct phenotypes, and clinical markers can help predicting functional consequences of novel GABAA-B2 variants. And from study 2, we concluded that transmembrane domain 1 proline variants across the different subunits of the receptor caused gain of function effect, and paralogs can be used for prediction of functional consequence of variants in important conserved regions. And how did we contribute to the personalized treatment? We did some clinical characterizations, functional characterizations, genotype-phenotype analysis, which are all the initial steps toward personalized treatment. And thank you for your attention.
Thank you, Dr. Mohammadi, for sharing your groundbreaking findings and explaining the phenotypes of the genes. I actually wasn't expecting that 50 million people worldwide are affected by epilepsy, and 70 to 80% were genetic epilepsies. So it's interesting to know that although the prevalence is low for GABA-A variant mutations, as genetic testing becomes more available, we'll discover that more people actually have these gene mutations. You know, a lot of our parents would like to understand whether their child is loss or gain of function, and knowing that functional testing is available is absolutely amazing. I was wondering if you could explain what and how functional testing is performed.
Functional testings, especially when it's ion channel we are working with, so performing electrophysiological recordings, is one of the ways to characterize the variants as gain of function or loss of function. And in our study, we used GABA sensitivity as one of the parameters. And in the second study, we identified it as the strongest parameter to characterize this variance function.
How do they actually do it in the lab?
The way we do it in the laboratory is that we have our cell models, which are the oocytes, and they are injected with the RNA from mutant receptors and the wild type receptors. So they take their time. Within 1 or 2 days, they are able to express the mutant and wild type receptors in the cell surface. Then we can apply GABA with different concentrations and then measure peak response of the receptors to the GABA. And this way we can see how sensitive is the receptors, mutant receptors, compared to the wild type receptors.
We have a question from Lou, and it's regarding the oocytes. He says that you were using oocytes as a cell line in your studies. How would the outcomes differ, if at all, if you used human cell lines?
That's actually a very interesting question. Of course, frog eggs they are not representing our human body and our brain physiology. And the next step would be to move on to human cell lines, iPSC neurons, and other type of cell models. All sites have also their own advantages, such as they don't have so many receptors by themselves on the cell surface, so it's not interrupting our results, which are the initial steps toward characterizing these variants and their function. So having more sophisticated cell lines is the main goal.
I wanted to share with you that we are working really hard to get biosamples into a biorepository. So we're collecting biosamples from our community, and hopefully in the near future, we will have enough biosamples where you and other researchers will be able to take them and create cell lines. I have a question for you about gain of function. Can you explain what you meant by low and high shift gain of function?
So when GABA was applied to our cell models, they were responding differently compared to the wild type receptors. So some the mutations, either loss of function or gain of function, were affecting the way that receptor would respond to GABA application, either by increasing the dose response curve or shifting the dose response curve to right or left. This way we know that it's increasing or decreasing the sensitivity of the receptor to GABA. And when I said that some of the gain of functions were divided into high shift gain of function and low shift gain of function, I meant that the magnitude of the change in GABA sensitivity was different. Some of them were shifting it from 0 to 5-fold change, and some of the others were changing the GABA sensitivity with higher fold shift, which was from 5 to 9-fold shift. So the magnitude of the change in GABA sensitivity was different, and the reason that we considered this difference was that when we're looking at the patients and their phenotypes, we could see that patients having high shift gain of function were having different phenotypes compared to the low shift gain of function variants. We could already see some differences. That's why we divided it into high shift and low shift. So I really think that we need to expand the cohorts and do more functional testings to be able to say more details about each cohort, but especially gain of function and loss of function mixture cohort, because it is a complicated one.
Do you have a grant that would help you with doing more studies, expanding that study?
Right now, actually, not really. I don't. I'm still working with it within the research field. I really would like to have more money to be able to perform more testings, especially creating maybe animal models as well, where we can explain how a gain of function can cause epilepsy or other disorders that we were seeing in my study. So there's a lot of interesting experiments that can be done, but right now I don't have funding, but I'm working on it, and hopefully I can come back to it.
We're coming together as a community in a project called Classy, where we're going to start a fundraising campaign. So hopefully this is something that we'll be able to contribute to you. I have a question from Mia, and she wants to know, were you able to find out which anti-epileptic drugs should be used or which to avoid for treatments of patients with gain of function variants?
That's also an interesting question. We have looked at our cohort and tried to understand whether which treatment was suitable for each cohort, but since our patients were treated with different medications at the same time, so we couldn't conclude which drug had the best effect or which drug caused more side effects than benefits. So that's something that we have to work on later and collect more precise data and ask clinicians to make like clear evaluations on the treatment and treatment response. But especially when it comes to GABAergic drugs for the gain of function patients, which is also suggested to be avoided for the gain-of-function variants in GABAA-B3 gene. So I'm assuming that's the case for GABAA-B2 gene patients, but based on our study, we didn't have clear answer for that. So that's something that we need to work on.
And we have one last question that just came through. Is having microcephaly an indication that a child has a gain of function? Have you ever seen microcephaly in loss of function?
That's actually so far, based on our study, it's a feature that we only saw in the gain-of-function cohort, actually. So it belongs only to gain-of-function variants and patients carrying this type of variants. We didn't observe it at all in the loss-of-function cohort.
Thank you, Dr. Mohammadi. Your work is such an incredible addition to the GABA-A variant mutation work. For us parents, understanding whether our child has a gain of function or loss of function is extremely important, as it affects our child's future planning, which drugs work best, and so on. The way that you explained that severe intellectual disability is associated with gain of function and walking and talking is associated with loss of function variants makes it so much more easier for us parents to understand. So I thank you again. We are so excited for all the future discoveries that you will make, and we are grateful that you are working on GABA-A for our children.
Thank you so much for having me, and see you. Thank you.
Bye.
Bye.